Got it. All right, let's get started. So, our speaker today, Dr. Adam Spira, has spent his career studying something that we all do every night: sleep. He's a professor in the Department of Mental Health at the Johns Hopkins Bloomberg School of Public Health with a joint appointment in psychiatry and behavioral sciences. He explores how the quality of our sleep shapes the aging brain, from day-to-day function to long-term risks like cognitive decline and dementia. So he connects the quiet hours of sleep to the broad landscape of brain health using tools from psychology, epidemiology, and neuroscience to uncover what happens when our nights go wrong. So I think this is something that we're all interested in, especially research-wise, and to see how that affects our memory, mood, and resilience to disease. So even though he's been here at Hopkins, sorry, excuse my cough. He's been here since 2008. It's long before I joined. So it's a little embarrassing that I'm only just now getting to know him. But I think in one of our early conversations, when I asked him if he knew someone else, he reminded me that this is a very large university. And even just the School of Public Health has, I think, over 800 or 900, some crazy number of faculty members. So I think it's okay and we're making progress. When I first started working in sleep with the Dr. French and Kathleen Weber, I was asking around for local experts that I could get input on. And when I asked her own Dr. Gamaldo, Adam was one of the first people that she pointed me towards. And then, just by chance, not long after, with the renewal of the MWCCS, the MaxWise Combined Cohort Study, Todd Brown also brought him in for his sleep expertise. So now Adam is regularly joining our sleep and neuropsych working groups, and I'm really excited to see how he's bringing his deep understanding of sleep and brain aging into the study of HIV and mental health. And while today's talk might not be focused on the HIV aspect, I think it's tools that we can all use. So today we'll get a glimpse of that broader perspective in a seminar. It's called Sleep, Aging, and Brain Health. So please join me in welcoming Dr. Adam Spira. This is where you would normally get applause, but it's on Zoom and most people have
Oh
their
no, I
cameras
see
off.
some. I see
Yeah,
some.
you can see the applause and you can
All good. And
please
thank you. What a wonderful introduction. I did not expect that. So thank you. Wow. What a good start. So yes, I will be, and as I've said, I've got too much to say, so I'm going to just dive right in. Hopefully you see the full screen version of this. I got a couple of, okay, very good. Thank you. So these are my disclosures. And I'm going to start by saying that something that you all know probably, that the US population is aging, and you're all aware of this. HIV shares lots of parallels with aging, as you all know, so I'm going to skip right through this. But what does the fact that the population is aging have to do with sleep? Well, sleep disturbances are common in older people. And poor sleep is increasingly being linked to poor health outcomes in older adults, including cognitive decline and functional decline and Alzheimer's disease. And because sleep disturbance is often very treatable across the life course, people are wondering, well, could it be a modifiable risk factor that we might be able to leverage to prevent poor later life health outcomes? So sleep related symptoms are really common in older people. In a survey of over 9,000 people from three communities, all older adults with a mean age of 74, more than 80% had a complaint sometimes or most of the time related to some aspect of their sleep. And when they looked at complaints that people endorsed as having most of the time, almost 60% had at least one chronic complaint related to their sleep. And why might that be? Well, some of this we believe actually is due to what we are calling, for lack of a better word, normal age related changes in sleep. What do I mean? Well, if you study people that appear to be free of some of the medical comorbidities that so often accompany aging, we still see in healthy people changes um, in sleep. And what am I talking about? Well, for example, sleep gets shorter as you move across the life course. And other noteworthy changes, as you look here, WASO is wake after sleep onset. So how much time you're spending awake after you initially fall asleep. So in addition to getting shorter, your sleep becomes more fragmented. You can see that you spend more time awake after initially falling asleep at night as you get older. We also see significant changes in sleep stages. You see an increase in the lighter stages of sleep and a decrease in, notably, a significant decrease in slow wave sleep, which is um, the deep restorative sleep that we believe is really important for memory consolidation and potentially for um, the prevention of Alzheimer's disease. And I can talk more about that. I must confess that I left a lot of the mechanistic stuff out because I figured we could talk about it in the discussion. But REM sleep also decreases over the life course. So those are things that happen even in normal people, we think. And in healthy people, actually, most of these changes occur by age 60 or 65. The only one that continues to change after age 65 is sleep efficiency, which is the portion of time in bed spent asleep, which actually continues to decrease as we move into later life. And so but those normal age-related changes in sleep are not, we believe, sufficient to account for the very, very high prevalence of sleep-related symptoms or complaints in older people. We think those are attributable to the medical and psychiatric disorders and medications that are taken for those conditions that people can accumulate as they get older. Also, decreases in physical function uh, due to disabling conditions, we believe can make it harder for people to get up and about and to exercise. And we know that exercise is good for sleep. So decreases in physical function can worsen sleep. Um, some of my research is actually focused, I should say, on, and there wasn't time to fit it in here, but looking at the other direction, does poor sleep predict decline in physical function and physical capacity? And the answer is yes. Psychosocial changes as people get older, such as retirement, where one's schedule might become somewhat less structured, taking on caregiving responsibilities for spouses, uh, parents, you name it, grandchildren. Those things can make a difference and interfere with sleep. And then bereavement becomes more common as we get older, as we lose more people that we've potentially known for many years. And so all of those things can have effects on sleep. We also know that so what are circadian rhythms? First of all, these uh, endogenous rhythms that and many aspects of our physiology oscillate on this roughly 24-hour cycle. Roughly 24 hours, circa almost diem day, so roughly daily. Those things include things like core body temperature, hormone production, um, gene expression, and the sleep-wake cycle is perhaps the most salient expression of the circadian rhythm that we're all aware of. Um, and So as we get older, we tend to see what we call a circadian phase advance, such that people get sleepier and go to sleep earlier in the day, and then wake up uh, earlier in the morning. And that can lead to sleep complaints as well. When people say, "Well, I'm waking up at 4:30 a.m. Why is that?" Well, if you're going to sleep at 8:30 p.m., it might not really be a problem in terms of total duration, but it can lead to some concerns. And then sleep disorders can also really affect people's experience and lead them to endorse um, sleep symptoms. So, for example, sleep disordered breathing is very common, and we'll talk about that more a bit later. So, when I came to Hopkins, I first was doing work. I had a K award look from NIA focused on sleep disturbances and functional decline. But I was also really interested in sleep and Alzheimer's disease. And so I'm going to switch to talking about that now. So, as you all likely know, Alzheimer's disease is the most common cause of dementia. It's characterized neuropathologically by extracellular beta amyloid or A beta plaques early in the disease course. And then later in the disease course, we see intraneuronal neurofibrillary tangles as the other major hallmark. In 2024, 7 million adults age 65 and older had AD, and the prevalence will quadruple from the year 2000 to 2050 globally. And there's no cure, so prevention is really critical. When people have thought about the links between neurodegenerative disease broadly and sleep-wake disturbance, this is the way that they have typically thought about it. That the brain changes, and that leads to disturbances in sleep and wake. And actually, what we see in people who have older adults with Alzheimer's disease and related dementias, when we compare their sleep to the sleep of older adults without those conditions, we see essentially an exaggeration of the normal age-related changes in sleep that I mentioned earlier. We see that sleep is shorter and more fragmented, just as we see across, as if you look across the life course in cognitively healthy people, right? And then lower sleep efficiency, spending a smaller proportion of time in bed asleep, reductions in slow wave sleep and REM sleep, and greater dementia severity is linked to greater sleep fragmentation. And we think this happens due to a loss of brain volume in regions that are critical for um, sleep-wake regulation, including in the suprachiasmatic nucleus. So the SCN, as it's known, is the central clock in our brain in the anterior hypothalamus, and it coordinates the clocks that exist in every cell of our body peripherally. And we know if you look here, you can see a very old slide, but you can see that the number of the volume of the SCN decreases somewhat across the life course. And then compared to older adults without dementia, you can see that those who have dementia have significantly less volume, brain volume in that region. The intermediate nucleus is also affected. So that's one mechanism. Another one is reduced exposure to bright light. Bright light is the key environmental cue, or Zeitgeber is the German word that's often used, that synchronizes our sleep-wake cycle with the external environment. And if you're not getting enough exposure to bright light, perhaps because you are in a nursing home, or you have you have dementia and you're at home and you're not going out and being as active, that can lead to more sleep-wake disturbance. Also, reductions in daytime activity and more napping uh, can lead to more napping, and more napping depletes the homeostatic sleep drive that builds from the time that we wake up in the morning until we next sleep. And so if you're napping, you're depleting it, and then at night it might not be enough to lead to, it might not be strong enough to drive you your sleep to through the next morning. And there are alterations in the cholinergic system that also play an important role in both Alzheimer's disease and sleep disturbance. We've seen these, so I described the sort of clinical presentation of sleep in Alzheimer's disease, and this has been shown experimentally in animal models. So in an AD mouse model, Rowe et al. From Dave Holtzman's group at WashU showed that as amyloid plaques accumulate in the brain of these AD mice, the sleep-wake cycle is altered, and that if you perform active immunization with A beta 42 in these mice, it prevents the deterioration of the sleep-wake cycle. So this all supports this link from neurodegenerative disease to sleep-wake disturbance. In 2009, there was a really critical seminal paper that sort of turned things on on their heads and really led myself and many of my colleagues to pursue a whole other line of research, which was Kang et al., also from Dave Holtzman's group at WashU, showed in an AD mouse that if you sleep deprive the mouse, you get substantially greater plaque burden, A beta plaque burden in the brain. And that's what you see here in the dark bars representing the sleep restricted mice, and the white bars representing the platform controls. And then my colleague Mark Wu, he's senior author on this paper. We received a grant from the Hopkins Brain Sciences Institute to study sleep and A beta in humans and in fruit flies, in Drosophila. Mark does a lot of Drosophila work, and now increasingly mouse work. But he showed in these flies that if you shake the little tubes in which they reside during the day when they're awake, they are like us, they sleep at night, unlike mice who sleep during the day. But if you shake these tubes during the day when the flies are already awake, you get this much. If you look at the bright green, that's an immunostain for A beta. Uh, you get that much A beta deposition in these AD flies. If you shake it at night when they're supposed to be sleeping, you get much more. So these studies really led people to think and other studies, these are just two, is there actually a link going in the opposite direction, not necessarily from neurodegenerative disease to sleep-wake disturbance, but might it also work the other way? And this started a whole program of research that, I'm just one of many people who has really uh, uh, taken to this area. And so in collaboration with NIA scientists at in the intramural research program, um, we performed a study of 70 people in the BLSA who were age 76 on average, highly educated, as is the case with the Baltimore Longitudinal Study of Aging. They gave self-reported sleep variables and then had an amyloid PET scan with Pittsburgh compound B within 5 years subsequent to that um, self-reported sleep. And we found that those who reported shorter sleep duration actually had greater A beta deposition after accounting for a range of potential confounders in the cortical distribution volume ratio, which is looking at global sort of multi-region measure of amyloid deposition, and in the precuneus, which is a region in which we see A beta early in the AD course. We saw a statistical trend towards a link between trouble falling asleep and A beta deposition, but nothing significant. But we did find that those who reported that their sleep was more restless had greater A beta in the precuneus. And then this is showing just going from the longest, people reporting the longest sleep to somewhat shorter sleep to the shortest sleep on the right-hand side, that as you move from longer to shorter, you get more A beta deposition. Sticking with the A beta theme, I found that in the BLSA, they had way back in like the 1990s through early 2000s collected some additional sleep measures that we could then link to A beta subsequently. And one of them was a question, do you often become drowsy or fall asleep during the daytime when you wish to be awake, e.g. falling asleep watching TV or reading? And so we called that EDS, or excessive daytime sleepiness. And in 123 people, we compared those who were negative for EDS to those who were positive. Those who endorsed that they were falling asleep a lot, after accounting for several potential covariates, we found that that EDS positive status was associated with 2.75 times the odds of having a positive amyloid scan, an average of 16 years later. We thought that was pretty remarkable. We published that in the journal Sleep. And we've continued to do a bunch of work in related areas. I want to talk a little bit about sleep disordered breathing, which is characterized by repeated interruptions in breathing during sleep. If you stop breathing, it's an apnea. If you have significant reductions in your airflow accompanied by blood oxygen desaturation, and depending on the definition, and or an arousal from sleep, then it's a hypopnea. Obstructive sleep apnea is the most common type of sleep disordered breathing, and the risk factors include age, uh, higher BMI, greater neck circumference, and male sex. It's measured by polysomnography, and the most common metric that we use is the apnea-hypopnea index, which is the number of apneas plus hypopneas per hour of sleep. And SDB, as it's known, is highly prevalent in older people. Depending on where you look, it could be about 20% of people aged 60 to 99, but then in other studies, as many as 62% of adults aged 65 and older. When I was a postdoc at UCSF working with Christine Yaffe, this was the first study I led with that group, and we showed in the study of osteoporotic fractures, a mostly white sample of older women who were age 83 when they completed polysomnography and some cognitive tests, that we looked at the MMSE and scores that were more than 1.5 standard deviation below the mean, and we saw significant association, so that for every standard deviation increase in the AHI, there was a 40% greater odds of being impaired on the MMSE. Severe sleep disorder breathing was associated with almost 3.5 times the odds of impairment. If your blood oxygen saturation dipped below 80% at any time, you had 2.7 times the odds. So we saw a bunch of associations there. And this is the central apnea index, which refers to apneas that we believe are mediated by the central nervous system rather than by an obstruction. We didn't see anything for Trails B, but that might be because there was no correction for Trails A. That's a whole other thing. But we also looked at genetic moderator. ApoE is one of the, it's probably the largest genetic risk factor for Alzheimer's disease. And when we looked at ApoE negative folks and ApoE positive folks, we saw that the association between apnea-hypopnea index and impairment on the MMSE was much stronger in those who were ApoE4 positive. Very broad confidence interval, but and a 0.08 p for interaction, but we called it an interaction nonetheless. And then a few years later, I was fortunate to be part of this paper that really became a seminal paper on sleep disorder breathing and dementia that Christine Yaffe led in 300 women in the study of osteoporotic fractures. And we called sleep disorder breathing positivity if you had an AHI of 15 or greater, which is really moderate to severe sleep disorder breathing. And we saw that compared to those who did not have that, those who had moderate to severe sleep disorder breathing had 1.9 times the odds of having MCI, mild cognitive impairment, or dementia 5 years later. And we saw that in some analyses looking at whether it was because sleep disorder breathing does two things to you physiologically. First, it fragments your sleep. Secondly, it leads to hypoxemia. So we tried to disentangle the extent to which it was the fragmentation or sleep loss versus hypoxemia, and it really appeared to be the hypoxemia that accounted for it. Moving back to away from sleep disorder breathing, the reason I talked about that now is excessive daytime sleepiness, or EDS, is that I showed you the study looking into amyloid deposition later on. One of the big causes of it can be sleep disorder breathing. So that's why I switched to sleep disorder breathing. But I want to switch back and talk about it just to show you another study that I did with my colleagues at NIA and 122 participants in the BLSA neuroimaging initiative who were 67 when they reported their average sleep duration. And then they had an average of 7.6 1.5T MRI scans over 8 years on average. And we looked at repeated measures of cortical thickness, and we only used data from visits in which participants were cognitively normal. And we found that when we compared sleep duration, our reference category was 7 hours of self-reported sleep. Those who reported fewer than 7 hours had more rapid cortical thinning in frontal and temporal region, these frontal and temporal regions, almost all in the left hemisphere. We're not sure why. And then interestingly, we often see a U-shaped association between sleep duration and poor health outcomes. And here we also saw that if you compare people who had longer sleep than 7 hours, you actually saw more rapid thinning in these frontal regions. This is a pretty brain picture showing these regions. I want to point out that these people remained, they do very, very rigorous cognitive testing with adjudication of cognitive status through the Hopkins ADRC in the BLSA. And these participants remained cognitively normal throughout all of the MRI scans. But we still saw these associations. So linking sleep, various aspects of sleep to various aspects of brain health and cognition. I want to shift gears now to talk a little bit about circadian rhythms. So as I mentioned, these are aspects of our physiology that follow a roughly 24-hour cycle controlled by the SCN, which you see here in green, just above the optic chiasm. That's the suprachiasmatic. It's referred to the optic chiasm. And you can see here that from the optic nerve, it's innervated by the optic nerve. And that's it works out well because it turns out that light is the central or the principal Zeitgeber or synchronizer with the external environment. And we can measure these circadian rhythms in various ways, including through actigraphic movement data, so wearable sleep trackers. And there are various ways that you can quantify rest activity rhythms. They're illustrated here. You can look at peak to trough distance or amplitude. You can look at acrophase, which is the timing in the 24-hour period of the peak of the rhythm. You can look at the mesor, which refers to the mean estimating statistic of rhythm, which is sort of like the rhythm of the mean of the rhythm. And we have non-parametric indices that don't see. This is based on the assumption that there's a cosine or function or shape to the rhythm, but we also have non-parametric indices that don't rely upon that assumption. People use a bunch of different other metrics to get at circadian rhythms. I'm going to show you some results that I think are fairly interesting. Now, we did a study in 2021 looking at people who are A beta positive but cognitively normal, and A beta negative and cognitively normal in a relatively small cohort from the A4 and Learn trials of solanezumab. And we found that actually, when we looked at these cosinar and non-parametric indices, these conventionally used circadian rest activity rhythm metrics, we found no difference. And we thought, well, it could be the sample size. It was relatively small. But we used a new method, a data-driven method, and it was exploratory in that first study that was published in 2021. And we saw some differences. And I thought, well, we really have to replicate this in another cohort. So we did in the BLSA. So in the BLSA, you can see the accelerometer we used. From 2012 to 2021, 560 individuals provided 765 observations or wore, the device for 765 periods. And they were asked to wear it for 7 24-hour periods to complete a sleep diary and press an event marker button so we could score the data. And we have data in BLSA available on sleep, 24-hour rest activity rhythm metrics, and we do have available light exposure data that um, uh, we have to get that to the NIA. But I mentioned this other metric that we used in this other study that was published previously, and it came from this method. I collaborate with great people in biostats, including Vadim Zapunikov, who was a co-author on this paper. And what we ended up doing is using function on scalar regression, in which the predictor was in this cognitively normal set of participants in BLSA, primary predictor was A beta status. The outcome were these 24-hour activity profiles. We adjusted for age, sex, and education. And there are two ways. Well, I'm going to move forward because I don't want to run out of time. But what you'll see here is, so on average, these people were 76, about half women, just over half women. 32% were ApoE carriers, 76% white, highly educated, and they were all cognitively normal. What we found using this method, if you look across this 24-hour period, the blue refers to periods in which there is a significant difference between people with A beta versus those without. PIB, Pittsburgh compound B positive versus negative. And what we found is in this afternoon period, right? So let's see, when we used the more conservative method, there are two different methods to establishing p-values when you use Fossor. One is more conservative and one is less conservative. The blue region is the region of where there's a difference per the less conservative method. The pinkish salmon regions, that refers to the more conservative estimate of a difference. But we found that compared to those without A beta, those who had significant A beta deposition had higher mean activity levels across days between 1:30 p.m. and 2:30 p.m. And so this was nice for us to see because this is what we had published in the other article. earlier in 2021, we had shown the same difference in the afternoon. We'd seen some other differences too, but this is the one that we were able to replicate. We also looked at differences in day-to-day variability in motor activity in adults with and without preclinical A beta deposition. And one of the areas that we saw that was replicated from the prior paper was lower variability in activity levels in this afternoon period. You can see it again here from the prior study. So the question of this was a cross-sectional study. Both of these have been. So the question is, could it be that circadian rest activity rhythms, or at least using this Foster method, could serve as some sort of a marker of neurologic change? In this case, one specific to Alzheimer's disease, but could provide a window on the on brain health, potentially across various neurodegenerative conditions. Um, there's also reason to believe, and we've published papers looking at circadian rest activity rhythms predicting change in cognition. We've shown that that is indeed the case, and that these rhythms, the more conventional rhythms, we have a paper that we're resubmitting soon that actually shows that these rhythms predict change in MRI derived brain volumes, and that those associations actually differ by age. So stay tuned for that one. I'm going to move to the last part of the talk. I'm actually doing okay on time. I'm shocked to look at sleep wake and biological aging. And this first study, which has not been published, it's being presented. I think this one, or maybe we're submitting it for a conference, and she's working on the paper. But Cassandra Nijhuis did uh, her master's in the Department of Mental Health. Then she went and got a PhD at Penn State in sleep epidemiology, and she's now a postdoc working with me and Dr. Alden Gross in epidemiology. So, and this study has got links of multidimensional sleep health with epigenetic age acceleration. So I guess a good question for you is, what characterizes good sleep? Any thoughts? Does anyone, how would you describe good sleep? Feel free to come off mute if you're brave enough. Anyone?
No interruptions during the night.
No interruptions. So consolidated, continuous. Right. So I like that. What else?
Waking up feeling like you're fully rested.
Feeling rested upon awakening. Yes. Anything else?
Don't have a lot of dreams.
Oh, interesting. Dreams, which mostly occur during REM sleep. Yeah, and then other things, is it of appropriate duration, right? Because you can have sleep that is really consolidated, but if it's of an and continuous, but if it's of short duration, it's not enough of a good thing, right? So the point going through this, there are various things that can various ways in which we can think about sleep health. And there's a relative over the last 10 years, maybe 11 now. Yeah, 11. Look at that, 2014. This idea of multidimensional sleep health has really emerged as an alternative way of looking at just isolated sleep duration, and then looking separately at sleep fragmentation, and then looking separately at sleep satisfaction. And there's a model called Are You Sated that assesses the six dimensions of sleep health, and this is increasingly used. So the R refers to regularity, the consistency of your bed and wake time. The U is just because they had to have a good acronym. I mean, that's the most important thing. But are you sated? The S is for satisfaction, your subjective assessment of your sleep. Was it of good quality? A is for alertness, your ability to maintain attentive wakefulness during the day. T is for timing. When in the 24-hour day is your sleep happening? Sleep at different times of day can have implications for health. If you are sleeping at a time that is out of sync with your circadian clock, you actually are negatively affecting your health potentially. And that's why we think shift workers have so many poor health outcomes. So that timing is important too. Efficiency. the ease of falling asleep and returning to sleep, and duration, the amount of sleep that you've obtained. And what happens is you can measure these things various ways: self-report, objective measures, some like satisfaction, of course, you can only get by self-report. But you dichotomize each of these dimensions and give people a point for each of them for which they are positive and higher score. So you get a composite score of 0 to 6, where a higher score indicates better sleep health. Okay. And it's really, the literature has really exploded on multidimensional sleep health. We know that better MSH, as it's known, is associated with lower depression and anxiety, reduced cardiometabolic morbidity, lower healthcare utilization, less psychiatric distress, better physical functioning, and more physical activity. And let's see. But to date, I'm going to skip forward here. No studies had investigated MSH in relation to biological aging. So while chronologically we all are aging at the same rate, biologically some of us are aging more rapidly than others. By the way, oh no, that's fine. So there are various ways to measure biological aging. Telomere length has been around for a long time, for example. P16 is a marker of cellular senescence. But epigenetic clocks have really become quite popular. And these clocks, as many of you know, leverage patterns of methylation to quantify aging. And I'll discuss those clocks more momentarily. This curve here in this figure from Horvath and colleagues shows the population average methylation age as a function of chronological age. And if someone's biological age is higher than expected for their chronological age, that's accelerated aging, right? You're up here above the curve. And we all want to be this individual who is in the green, indicating that their biological aging is lower than their chronological age, which we call age decelerated aging, right? And these epigenetic clocks are based on patterns of DNA methylation, as I indicated, at different CPG sites. And they have really been well studied. And there are two generations. The first generation was designed to predict chronological age. And so those are Horvath and Hannum, those clocks. And then the second generation were captured to develop health risk and functional decline. And you see those here. And then grim age is one that's predicting mortality and lifespan and health span. So we wondered, what was the association between multidimensional sleep health and accelerated biological aging measured by epigenetic aging clocks? And so we studied this. And I'm going to skip in, let's see, how many parts is it? 512 older adults in the Baltimore Epidemiologic Catchment Area study, Wave 5, of which I was PI with Bill Eaton in the department. I'm now PI of Wave 5.5 with Brian Mayer in my department. And Brian is a statistical geneticist, and so he's particularly interested in this stuff. They're age 55 to 99, a mean of 67. And so we came up with cutoffs for each of these things based on different sleep measures. We combined actigraphy for efficiency, sleep efficiency, and for sleep timing and duration. And then these were from self-report, these other metrics. So we had 229 adults after excluding people without methylation data and without actigraphy data. We adjusted for age, sex, race, the amount of physical activity people were getting using actigraphy. And in a second model, because smoking can play such an important role, we used a standardized polyepigenetic smoking score to additionally adjust. So I should note that we use, instead of directly using the difference between biological age and chronological age, we used the residuals from regressing biological age on chronological age. And so what do we find? So the mean multidimensional sleep health score was 4.19. And so about a third of the participants were below 3, which indicates that they were good on only half of the multidimensional sleep health domains. But what do we find? we found that, and I'll skip to model 2, which includes the smoking polyepigenetic score, that better sleep health was associated with slower grim age acceleration in both models. We saw this trend level association for slower Horvath age acceleration. So this suggests that better multidimensional sleep health may slow biological aging. We have to learn much more about this, though. I want to very quickly go over this last study led by Chun-Yu Liu, who is a doctoral student working with me and Brian Mayer. Rest activity rhythms are linked to many poor health outcomes if they are dysregulated, including cardiovascular disease and cancer. And some studies have linked them to proteomic clocks. Chronic circadian misalignment measured by long-term shift work has been linked to greater epigenetic age acceleration, but relatively few studies have examined rest activity rhythms from actigraphy with epigenetic outcome clocks. So we did that and examined whether they're modified by sex, given we know that there are sleep-wake differences in men and women and potentially in epigenetic aging too. And it's in the same cohort of people. We looked at relative amplitude as a rhythm strength measure. These are non-parametric rest activity rhythms indices, rhythm regularity, interdaily stability, and rhythm fragmentation, intradaily variability. And we adjusted for age, sex, race, education, smoking, alcohol use, et cetera, et cetera, et cetera, lots, and then did some stratified analyses. You can see some male-female differences. And I'm going to move on here to show you the interesting results. So what we saw for relative amplitude, which is a measure of rhythm strength, is that higher levels of relative amplitude were associated with epigenetic age deceleration according to the grim age clock, as you see here, and greater interdaily stability, meaning between days, how similar are your rhythms? Greater stability there is associated with age acceleration, a deceleration as well. This was epigenetic age deceleration here too, for grim age and for pheno age, which was trained on a bunch of medical comorbidities. We also saw that greater fragmentation of the rhythm, instead of having a nice smooth shape, having a more less rhythmic shape, was associated, that's intradaily variability, was associated with accelerated aging according to the grim age clock. We also found that this top panel shows men and the bottom panel shows women, and we found some interesting differences in the results. You can see here the blue, by the way, is indicating they're not crossing the null value. So those are the significant results. That while we saw that greater relative amplitude was associated with deceleration among men, it was not for women. We saw that, and that was for grim age. Similarly, for grim age, interdaily stability, which is regularity of the rhythm, was associated with deceleration in grim age in men and with pheno age in women. And nothing for intradaily variability in men, but it looked like women were particularly vulnerable to adverse effects. I'm using that loosely. It's not causal, right, necessarily, in grim age and Horvath. So that was interesting, showing basically that lower strength and stability and higher fragmentation are associated with greater epigenetic age acceleration with differences by sex, pointing to the potential importance of maintaining strong and stable rhythms to mitigate age-related biological deterioration. So in summary, sleep duration and quality, circadian rest activity rhythms, and sleep-disordered breathing are linked to key brain health metrics in older adults. Multidimensional sleep health and strong, stable rest activity rhythms are tied to slower biological aging. Does improving sleep and rhythms improve brain health and slow biological aging? We don't know. We would like to know. And to do that research, and then do combined. Oh, that was a separate one. That was a bonus. I didn't show you that study, but I want to show, before I say thank you, I want to show you this is the conceptual framework guiding ECA Wave 5.5, which is really looking at an infectious etiology of Alzheimer's disease, looking at herpes viruses, Toxoplasma gondii, and the virus that causes COVID as risk factors for cognitive decline and adjudicated cognitive outcomes, blood-based Alzheimer's disease biomarkers, and biological aging markers in that same ECA cohort. And we're looking at we're looking at sex, AD risk genes, and stress exposures as effect modifiers. And now I'll say thank you to the NIA who's funded all of this work, and my collaborators and staff, Cassandra and Chanyu for slides, and to the participants in all these studies. Thank you. Well, I only left you 12 minutes.
Hey, it was more than you promised in the beginning, so I think you did great on time. And you got all of the information in there. That was really fantastic. And already the chat is full of
Thank you.
lots of questions and comments.
Uh-oh. Let me look. Yeah.
Well, I can read them out to you.
Oh, I see them.
And then
I
if
see
you want to unmute, do you want to read them yourself, or do you want me to read them to you?
Sure. So, yeah. So, Pilwali, there may be another sequel novel to Insomnia too. I'm not sure exactly what that refers to, but I'm happy to hear it. But I do know Gina Poe's work has been great. She does fantastic work on sleep-dependent memory consolidation. So, Dr. Matthew Walker's group, including Bryce Mander, who's now at UC Irvine, has his own group. They have shown that actually amyloid beta and aging appears to interfere with memory. Brain aging appears to interfere with memory consolidation as well. So yes, that has been studied. Malachi Kao mentioned reduced daytime sleepiness. And I see Jules is asking about kidney disease. Does it affect sleep? You know, it seems like everything tends to affect sleep or be affected by sleep. It's one of the reasons I like studying sleep. I can collaborate with pretty much anyone for that reason. So I wouldn't be surprised at all if it affected sleep. I can tell you that in people with more advanced kidney disease, just the timing of dialysis can make it hard to sleep. People have to get up and get to their appointments. That can really mess with sleep. I see something about sleep, circadian rhythm and metabolism. Yeah, we're doing a study now in the look ahead cohort, which is people with obesity or overweight, all of who have diabetes, type 2 diabetes, and have been followed for 20 years. And we're studying links of sleep disorder breathing and circadian rest activity rhythms with their cognitive decline and Alzheimer's biomarkers. So this is a great question that Barbara asked, right? So she's getting at the glymphatic clearance. So I told you that I skipped from the mechanistic stuff because a big question is, well, how is sleep disturbance and circadian rest activity rhythm? How are those things actually leading to Alzheimer's disease? There are two main theories of why sleep leads to Alzheimer's disease. The first, which has been around for a bit longer, has to do with something known as the synaptic homeostasis hypothesis from Tononi and Chiarelli. Some of you would be familiar with this. The notion is that we wake up in the morning, we go about our days, we navigate our environment, we're constantly learning things and building building connections between neurons. And synaptic strength builds, and it reaches a crescendo potentially by the end of the day. And what we know is that while neuronal activity we generally think of as a good thing, it can become, there's such a thing as neurotoxicity, right? And so excess neuronal activity has been shown to increase amyloid beta production. And we think the synaptic homeostasis hypothesis states that during slow wave sleep, we have a downscaling of all of those synaptic connections that are made over the course of the day. And that in the absence of slow wave sleep in particular, and you can have your slow wave sleep disturbed through many different mechanisms, through the absence of slow wave sleep, we have a failure to down regulate those connections, to downscale those connections. And so you get a net excess of neuronal activity that leads to excess A beta production. That's one theory, is for that basically, if you don't sleep, you have an excess of production. The other theory comes from Xi et al. From Megan Nadergaard's group at Rochester. That's what, here we go, where was it? Yes, what Barbara was referring to. How does aging and fragmented sleep affect lymphatic clearance of A beta and tau during sleep? So what Xi et al. Published in 2013 was a paper showing in mice that during sleep there is an expansion of the interstitial space, particularly during slow wave sleep, that facilitates the flushing out of toxic metabolites. And that this and basically it's mediated by aquaporin 4 channels and by glia. And because it's glia, they called it, and there wasn't a lymphatic system that had been clearly identified in the brain at that point, they called it the glymphatic system, gla for glia. And that, really during anesthesia, under anesthesia, and during sleep in mice, you see much more efficient clearance of toxic metabolites during use through that lymphatic system. It's still a lot more research has come out about it, but it's still debated. So to the extent that slow wave sleep truly does modulate lymphatic clearance, if it's fragmented, Barbara, it would have adverse effects. And I have not kept up on all of that literature, but that's the best I can do at this point. It's interesting. GLP-1 agonists and central sleep regulation. That's an interesting question. I can tell you my colleague Atul Malhotra at UCSD published a paper in New England Journal not long ago showing that GLP-1, those medications can have a significant effect on sleep apnea. And so we might see big changes in how sleep apnea is treated. But I don't know the answer to that question about central sleep regulation as a function of GLP agonists. Carl, you discussed that sleep disturbances have been associated with cortical thinning. People with HIV have been reported, which has been associated with CD4 nadir. Yes. Yes, I totally agree. And so this is one of the things and I've done like 2 studies in HIV, I think, but I'm very interested in this because it intersects so well with my interest in sleep, which is indeed disturbed for various reasons in HIV, and neurodegeneration, and now with this ECA wave 5.5 infection. So I think I don't know of any evidence of this synergy between sleep, HIV, infection, or perhaps it's viral load, or other aspects of immune function and cortical thinning. I don't know, but I'm very interested. Can I comment, Keiju, can you comment on whether sleep medications reduce the risk? Oh, I'm so glad you asked. So sleep medications in aging tend to be, the American Geriatric Society publishes a list of medications to be avoided by older adults, and sedative hypnotic medications are among them. They can lead to falls and fractures, and actually they've been linked to greater dementia risk. That literature, though, we need to know more about the dementia risk piece. One of the reasons that, and I will say, on the other hand, there's a new dual orexin receptor agonists, or antagonists, sorry, are a new class of sleep medications that have come out. And my colleague Brendan Lucy at WashU did a small randomized trial in healthy young and middle-aged adults. And he showed where he did continuous lumbar catheterization over 36 hours, took repeated samples, and showed that the drug, which is a sleep aid, significantly lowered A beta and tau levels in CSF acutely. And so there's a lot of interest, but that is a specific medication with a specific mechanism of action. There are many other medications, for example, benzodiazepines and so on, that we know have effects on sleep. The question is, it's not enough just to make someone sleep, we think, using whatever hammer you have, right? Pharmacologically speaking, to have good brain health outcomes, we think. I can tell you that I've talked a lot about slow wave sleep as being potentially beneficial for brain health. I get trazodone, a commonly used medication for sleep that is actually not a sleep aid. It's used off label for sleep. It's a not very effective antidepressant at low doses, but it increases slow wave sleep. And so Paul Rosenberg and Barry Greenberg in psychiatry, I'm a co-I on their grant, which is an RCT of trazodone for sleep, to see if we can improve next day function and cognitive performance over time. Arnold Bacher's on that project, looking at functional MRI and AD biomarkers over time as a function of boosting slow wave sleep through trazodone in mild AD and MCI. So these are great questions. Some sleep medications, depending on the class, may actually be beneficial. Some may be really bad news. Cannabis, Cecilia, I'm so glad that you mentioned that. I could go on for a long time here. We don't know enough about cannabis. Cannabis definitely is used by a lot of people for sleep. And and people often stay on it because they have really bad rebound insomnia when they stop using it for sleep. So that's a great question. And we know now that there's a tremendous increase in use of cannabis in older adults. Melatonin is an interesting one. Yeah. Uh, yes. So we don't know how all of these things. Yeah, this is a great question, Daniel, and Cecilia and Raha. I don't think we have time, but
All right, so for
you.
part 2,
Yeah.
we're going to talk about sleep interventions. I think that's going to be your next talk.
And mechanisms as
And
well.
mechanisms. Yeah, the
I-
mechanism stuff is really fascinating. I mean, there are really those two main mechanisms that people are interested in, glymphatic and this sort of synaptic homeostasis. Yeah. But great questions. Thanks for all the interest.
Really wonderful
that was
talk. Really
fantastic.
great.
Oh, thank you. Thank you.
And you
Thank you.
even got all the questions in it on time and addressed them all. That was
Amazing
really
job.
impressive.
Thank you. You guys are too kind. I'll come back.
Wow, you're getting off the bus now. All right, thank you. Caroline, do you
Thank
wanna,
you, Adam.
the speakers for
Caroline, do you
next
want to,
week?
I have to run, but could you mention the
yeah.
talk for next week?
Yep. So next week we have Anastasia Williams and Sebastian Molnar, and they're presenting a talk, The Intersection of HIV-1 and Extracellular Vesicle Research. So I hope you could
Cool.
join us then. All right. Thank you.
Thanks.
Thanks, everyone.
Bye.
Bye.
Thanks.